Hello everyone. Today I will be discussing about antibody functions. So we know that there are two types of immune responses, right? There are two types of immune responses. First is the cell mediated and second is the humoral response. So first cell mediated and second is the humoral response. So now in the humoral response, the effector molecules are the antibodies. Okay. So in antibodies, in the last lecture only we had seen the structure of the antibody. It was a Y-shaped structure and it consists of heavy chain and two heavy chains and two light chain. They both are linked together with the disulfide bonds. So here we know that the tip of the antibody contains FAB region and the tail of the antibody consists of FC region. So in the last lecture I have told you the function of both these regions, that from the term only we are get to know the function, that is fragment of antigen binding. That is the function of FAB region is it binds with the antigen. And FC region means FC region will interact with the FC receptor of the phagocytic cells so that the further it can activate the immune response. Okay, so these antibodies, they are the let me show you the structure. So this FAB and FC region, these both act together to drive the antibody effector function and pathogen clearance. How they are doing it? They are, suppose this is the antibody structure and they are linked by disulfide bonds, and here this is the FC region, right? And this is the FAB region. So this FAB region, it will be interacting with the epitope of the antigen. What is epitope? Epitope is the part of the antigen which is immunologically active and which is getting recognized and binded by the FAB regions of the antibody. So this is the antigen and this is the epitope. Okay, so here, and when this FAB region, when it interacts with the epitope of the antigen, the FC region will be interacting with the FC receptor of the phagocytic cell. This is the phagocytic cell. So this is the FC receptor. So when this FAB region is interacting with the antigen or the epitope of the antigen, the FC region will interact with the FC receptor and then of the phagocytic cell, and then the further signaling will occur in the phagocytic cell so that it can initiate the effector functions. Okay, so from here we can understand that how these FAB region and FC region both are contributing for proper functioning of the antibody and for further initiating the effector functions. Okay, so this was just the brief about the structure of the antibody and how their domains are contributing for the functions. So here there's a these are the functions of the antibody, that here, as I have told you, again the same thing is given here. Variable region is the FAB region and constant region. They are contributing for the antibody functions. Now FAB region, as I have told you, it will be interacting with the epitope of the antigen, and FC region will interact with the FC receptor of the pathogen. Okay, Now here we can see that it's shown 4 different functions of antibody. First is the neutralization. So here we can see that these antibodies, they have bound with these, they are neutralizing the pathogen. Actually this is some virus or pathogen or any kind of toxin, and they neutralize the pathogen by binding, and then they are blocking the receptors that the pathogen use to gain entry into the cell. Entry into cell. So therefore they are blocking the receptors so that they cannot enter into the cells. So here these are the antibodies, and they are binding there with any kind of viruses and toxin to neutralize them. Virus and toxin neutralization and further prevent pathogen host binding. Second is opsonization. Opsonization, and these antibodies, here we can see this is the pathogen, right? This is the pathogen, and these are the epitopes to which these FAB regions are recognizing. These are the antibodies. So antibodies are recognizing the epitope, and as I have told you that FAB will bind with the epitope, and FC region will bind with the pathogen or the it's not pathogen, it's phagocytic cell. I have written wrong here. It's a phagocytic cell. Okay. So FC receptor, this is the FC receptor, and here is the FC region. Now FC receptor of what? Of phagocytic cells. This is the macrophage. So after binding with the epitope, these antibodies are binding with the FC receptor, and they're binding and they're recruiting, and further they're initiating the phagocytosis process. Okay, so next is third is the complement fixation. How it is doing these antibodies, again they are recognizing, this is the antibody, and these antibodies are recognizing the pathogen or any kind of tumor cells, and further they are initiating the complement cascade. This is the part and this is the one of the component of the complement cascade. Okay, and then they are initiating the complement cascade, and further they will be involved, that complement cascade will be producing the MAC complex, and further the membrane rupture will occur. So here we can see that these are antibodies, they are recognizing, and further they're initiating the complement cascade. So complement fixation and formation of membrane attack complex. So what will happen after membrane attack complex? There will be lysis of the cell or phagocytosis of the cells. Now last one is the antibody dependent cell mediated cytotoxicity. From here we can see that they cooperate with cellular branch of immune system by binding and recruiting the activities of cytotoxic cells like NK cells. Here again these are the antibodies, and these antibodies they will recognize infected or tumor cells, and then again they will further bind, and then they will recruit these cytotoxic cells like NK cells, and then these NK cells will be releasing some mediators like granzymes, perforins to destroy the or to kill these infected or tumor cells. So then after that what will happen? NK induced apoptosis will occur. So in this way these antibodies are contributing in different functions. So next is, so let us now go in detailed explanation of every mechanism. That is, first, as I have told you that the first mechanism was neutralization. So neutralization, so from the term only we are getting to know that it must be using the antibodies which will neutralize the effect of the pathogens. So antibodies which neutralizes the effect of pathogen. Okay, so and these are produced naturally by the immune system, and how their production is triggered after, by both infection. It can be both infection or vaccination against the infection. Vaccination against the infection. So under these conditions, these neutralizing antibodies are produced. Antibodies are produced. Okay. So let us just get a brief about neutralization. That antibody binds to sites on the pathogen or toxin that interact with the host protein. Okay, so they are binding with the any kind of pathogen or toxin, and they will mask them, inhibit the entry of that pathogen or toxin into the host. Antibody pathogen complexes are then eliminated, often after phagocytosis. After, when they are masking and they are inhibiting the entry of those cells or those pathogens, then they will be eliminated by the phagocytosis. So next is, how do neutralizing antibodies work? So we can see in the diagram that this is one of the virus, or some of the we can also say toxin. So here the antibodies, they are recognizing, neutralizing antibodies bind to viruses. They are binding to the viruses, and after binding, they are blocking the pathogen interaction with the cell's receptor. So therefore there will be no receptor binding, and therefore no infection will occur. Okay, so now the neutralizing antibodies can neutralize toxins, they can neutralize viruses, they can neutralize microbial virulence factors. But this one is seen in vitro condition. Okay, so now let us see one by one neutralization of toxins. So how the neutralization of toxin occur? Here again in the diagram we can see that these toxins, they are recognized by the antibodies, and they have masked them properly, and thus these toxins are not able to bind to the host cell receptor, and further they will be blocked. So here many, like which bacteria or pathogens are producing toxins. Many pathogenic bacteria like Vibrio cholerae, Clostridium tetani, so all these bacteria, they can cause disease by releasing the toxins, right? And how these toxins are neutralized? They are neutralized by FAB domain of the antibody. They will be interacting with these toxins. Okay, and then they will block the binding with the receptor. With which receptor? Block binding with the receptor. Binding with the host cell receptor, right? Binding with host cell receptor. And further they will be preventing the infection. So in this way they are neutralizing, the antibodies are neutralizing the toxins also. Now neutralization of microbial virulence factors. This is seen in vitro conditions. Now these antibody, they can neutralize they can control the quorum sensing, they can regulate the biofilm formation also. So the first step in any colonization of the host for the commensal organism is the attachment to the mucus. Attachment to mucus, mucosal surface we can say. So mucosal surface. So what these antibodies do, for example, IgG antibodies has been found that these antibodies, they're specific for streptococcus pneumonia cell surface proteins, okay, and they will block the adherence of the bacteria to epithelial cells. Suppose this is the one of the epithelial cell, okay, and these streptococcal pneumonia, they might be interacting with the epithelial cells to enter into the cells. Now how these antibodies, they will neutralize, the FAB domain, the FAB domain will interact with these bacterial cells, and further when they will interact, they will be blocking their entry into the epithelial cells. So further they will not be able to enter. Okay, so this is not seen in, it is only seen in the in vitro conditions. Okay, so let me draw in a very clearer manner. This is the bacterial cells, and here this FAB domain is interacting, and this is the IgG, and here therefore these are not able to enter the epithelial region. Okay. Next is neutralization of pathogen entry and replication. Now some bacteria or viruses, this is seen in mostly virus, they will enter into the cell by binding to cell surface proteins by stimulating the endocytosis. Okay, now let me change the color. Now here we can see this is the virus, and it is in the, when the antibodies are absent. Here antibodies are absent. So what is happening in natural condition? The virus is entering into the cell. It is attaching to the host cell receptor. The attachment process is there, and then it will stimulate the endocytosis process, and further after endocytosis, the uncoating of the RNA will happen, and further the release of the RNA will be there. So in this way it is infecting the cell. In the absence of the antibody, there is an infection by the viruses or the disease by the virus. But when the antibodies are present, when the neutralizing antibodies are present, now what is happening? When these neutralizing antibodies are present, they are interacting with this, or they are neutralizing the virus by interacting. See, these antibodies are interacting with these viruses, and now because now this virus has been masked by the antibodies, now they are not able to attach themselves to the host cell receptor. They have blocked the attachment. First step, they have blocked the attachment. Second step, they have also blocked the endocytosis, and now these they are not able to stimulate the endocytosis, and further they have also blocked the uncoating of the RNA genome. So in this way, these antibodies, they have blocked the viruses in different what we can say, in different pathways, in different manners, right? In different ways it has neutralized the viruses, and it has prevented that viruses from entering into the cell. Now, escape from antibodies. So we know that even though the neutralization is most effective mode of protection against the infection, but still some viruses, they have developed mechanism to avoid the antibody mediated neutralization. For example, one obvious target for the neutralizing antibody, this is the neutralizing antibody, this is the neutralizing antibody, and their target is to bind with the viral attachment protein, any kind of virus attachment protein, so that they cannot enter or they cannot interact with the host cell receptor. So if these attachment proteins are interacted with these antibodies, they cannot interact with the host cell receptor, right? They are not able to interact. So what the virus do, some viruses, they will hide their attachment proteins. This is one of the mechanism by which they escape through the from the neutralizing antibody. They hide their attachment proteins, right? The amino acids which are very much important here, the amino acid which is very much important for the attachment, they hide these amino acid, or they will keep these amino acid within any kind of folds or clefts, and thus the antibodies, they are not accessible to these attachment proteins, right? And thus these viruses are able to invade the neutralizing antibody. So this is the case with the surface protein of, example is surface proteins of HIV, surface proteins of HIV. This is the first or second example is the HA protein of influenza virus. Also, in the second method they use, or the second mechanism they use from escaping the neutralizing antibody, is in HIV they escape by mutation of few amino acid. And when there is a mutation, that means they will be producing the genetic variants. And thus again they are able to evade the neutralizing antibodies. Okay, so these are some of the mechanism by which these viruses, they are trying to escape from the neutralizing antibodies, and thus they are able to cause the infection or disease. Next is opsonization. So second function of the antibody is opsonization. So we can see what is opsonization, the process of recognizing and targeting evading particles for phagocytosis. That is, the antibodies, they are recognizing and then they are targeting those foreign particles so that they can be processed for the phagocytosis. Now, what are opsonin molecules? So, opsonin molecules are the molecules that mark the foreign particles for phagocytosis. Now, there are two types of opsonin molecules. First is the antibodies, and second is the complement C3b. Now, why these opsonins are important, or why these opsonins are required? Why opsonins? Because phagocyte and cell wall of the pathogen, both are having negative charge. This is also having negative charge and it is also having negative, and therefore they will be, this negative charge makes them repel each other, and since they will repel each other, so they cannot be, this pathogen cannot be engulfed and it cannot be processed for phagocytosis. So therefore these opsonins are required so that they can overcome this process. They will overcome the repellent force which is present in between the phagocyte and the cell wall of the pathogen, and makes them and promote the uptake of this pathogen, and further they will be promoting the uptake of pathogen by macrophage or phagocytic cells. Okay, so that's why opsonins are required. So let us brief the opsonization process. Antibody binds to pathogen and is then bound by Fc receptors on the phagocytic cell. As I have told you, Fab region it will interact with the pathogen and the Fc region will interact with the Fc receptor on the phagocytic cells. Now the antibody antigen interaction or binding to Fc receptor, it will induce internalization and destruction by the phagocytic cells. So further there will be destruction by the phagocytic cells. Now as I have told you that there are two types of opsonins. First was the antibody and second was C3b molecules of complement system. So first let us understand how antibodies are doing the process of opsonization. So we know that here again the same thing I will be telling you, that it is having heavy chain and light chain, Fab region, Fc region. So Fab region it will interact with the epitope of the antigen, and Fc region will interact with the Fc receptor of any kind of phagocytic cell or macrophage cells. And when this interaction occurred, then after that there will be the, suppose this is the antigen and these antibodies they have coated them. So there will be, after interaction with the Fc receptor, this is the Fc receptor, no? There will be the process of engulfment and the phagocytosis process by the phagocytic cells. So what these antibodies are doing here, what is the function of these antibodies? They are increasing the chance, they increase the chance and efficiency in which the pathogen is engulfed. And further these pathogen will be destroyed by the lysosome. So in the diagram also we can see the same thing, that this is the Fc region. Let me change the color. This is the Fc region. This portion is the Fab region, and it is interacting with the pathogen, and the Fc region is interacting with the Fc receptor of any phagocytic cell. And after that, after the interaction of the Fc with the Fc receptor, there is a further signaling into the phagocytic cell so that it can stimulate the phagocytosis process, and here we can see the phagocytosis process is occurring by the phagocytic cells. So in this way the antibody is increasing the efficiency of the phagocytosis. Next is second molecule, second opsonin molecule is C3b. Yes, C3b is the second molecule. Now here again we can see that in the complement system, I will just tell you the brief about the complement system. The detail of this complement system we will discuss in another video. So here, complement system, actually it consists of many different proteins, around 30 proteins that improve the 30 proteins that actually improve the ability of the antibodies and the phagocytic cells to fight invading organisms. So this complement proteins complement system, actually it initiates the phagocytosis by opsonizing the antigen, and also they're responsible for inflammation and cytolysis. So just a brief about the complement system is that it consists of 3 pathways. Classical pathway, here we can see classical pathway, lectin pathway and alternative pathway. And these all pathways, they will be generating or producing C3b molecule, which will be required for opsonization process. So now let us start one by one pathway, how they are producing the C3b molecules. First is the classical pathway. Okay, now this classical pathway, it is an antigen antibody immune, it will form the antigen antibody immune complex. What will happen at first step, it will, in the antibody, this green color molecules, we can say this is an antibody. And these red molecules, it is shown here, it's the epitope. Now the antibody will first recognize the epitope, which is on the surface of the microbe, and these antibodies, when they interact with this epitope, there will be a conformational change in the Fc region, so that it will expose the binding site for the C1 molecule. Now this molecule, the complete, we can see here, this is the C1 molecule. Now what is C1 molecule? It's a part, or it's a component of complement system. Okay, now C1 molecule, actually it's a very large macromolecule. It's having 3 components inside it. It consists of C1q, 1 molecule of C1q, 2 molecules of C1r and 2 molecules of C1s. Okay, so the whole complex is of C1. Now here we can see this large portion is C1q molecule, and this blue ring like structure, it's a complex of C1r and C1s. Now C1r and C1s, they are actually a serine protease. I will not go in much detail now, I will just tell you in short. Okay, so here these are the serine protease. Now what will happen after, when this antibody, when it is interacting with the epitope, there is a conformational change in the Fc region, and that will expose the binding site for the C1 molecule. Now C1 molecule will bind there, and after when this C1 molecule, when it interacts with the Fc region, there is conformational change in the C1r, and that will get activated. It's another partner of C1, because it's having 2 molecules, no? So 1 molecule of C1 will get activated, and it will activate C1r, and both these C1r will activate C1s. Okay, now C1s, its substrates are C2 and C4. Now here we can see there is again one diagram has shown, and it has shown that it will be cleaving the C2 and C4 molecule. So C1 molecule will be cleaving the C1s molecule will cleave the C2 and C4. So we can see that it is cleaving C4 into C4a and C4b. C4b and and 2a, they both will combine together to form a C3 convertase enzyme. Now what is the role of the C3 convertase? We can From the name it is understood that it is converting the C3, that is inactive C3. Here we can see it is converting the inactive C3 into an active C3A molecule and active C3B molecules. Now the C3B molecule, as I have told you, what is the importance of this C3B molecule? It will be acting as a opsonin molecule, and thus we can see that it is used for opsonization, and C3A will be used for inflammation process. Now C3B molecule is produced in large amount. It's produced around 200 molecules are produced of the C3B molecule, and therefore some C3B molecule will be used for opsonization process and some will be binding or interacting with the C3 convertase to form C5 convertase. This is the C5 convertase, and further C5 convertase will cleave the C5 inactive C5 in C5b and C5a, and further MAC complex will be formed and the rupturing of the pathogen membrane will occur. So this was the pathway of the classical pathway, where how these antibodies are activating this initiating the complement cascade, and how they are all generating the C3B molecule, which will be used for the opsonization process. So this was the classical pathway. Now second is a lectin pathway. Now in the lectin pathway everything is similar to the classical pathway, but some differences are there. What are those differences? First is the MBL, mannose binding lectin. First, if we see in the initiation process in the classical pathway, we know that antibody was recognizing the epitope. Here the lectins, these are the lectin molecules. What are lectin? Lectin are the proteins which are recognizing the carbohydrate molecules which are present on the microbial surface. So this is the microbial surface and it is having carbohydrate molecule which the lectin will recognize. So here instead of antibody, lectin molecules are present for recognizing the microbial surface. Okay, so first difference is lectin, and then instead of C1 molecule, here again MBL molecule is present, mannose binding lectin. So for binding to the lectin, mannose binding lectin protein will be recruited, and again the same instead of C1 R2S2, which is equivalent to MASPs, MBL associated serine protease. Again this is the serine protease only. So instead of C1 R2S2, MASP to MASP are present. So this is the third difference. This is the second difference. Okay. So again they will be, the function of MASPs are also same, that it will also cleave the C2 and C4 into active parts and they will produce the C3 convertase. Again they will produce C3 convertase and that will produce C3B molecules. The same mechanism is similar to the classical pathway, only the difference was in this part. So this is how the C3B molecules are generated by lectin pathways also. Now last is the alternative pathway. Here the C3 molecules, there is a spontaneous hydrolysis of the C3 molecule and it will form a complex of C3H2OBB. Now this is acting as a C3 convertase. Again C3 convertase, the function of C3 convertase is to convert the inactive C3 into C3A and C3B. This is the C3B molecule. Again the factor B, D and properdin will bind and further they will form this complex C3B, BB, and this will act as a C3 convertase. Okay. In the classical pathway and lectin pathway, these were the C4B and C2A was used as a C3 convertase, but in alternative this C3b, Bb will be used as a C3 convertase. Again, the same C3b molecule will be produced. So from this we can see that all these pathway, they all are converging together to produce the C3b molecules. Now this C3b molecule, what is that? See here in the diagram we can see the C3b molecule, they will be interacting with the multiple sites on the surface of antigen. And then after binding with that, suppose this is the pathogen or this is the antigen, and here the C3b molecules, because it's an opsonin, so it will be binding with this antigen on the different sites on the surface of the antigen, and after binding with this, now they will bind with the receptor of the, suppose this is the receptor, this red one is the receptor, and then they will bind with the receptor also of the phagocytic cells, and this is after binding with these receptors, these are able to engulf, or there is a further phagocytosis. So these are the Fc receptor, and these are the, not we can say Fc receptor, any receptor on which the C3 molecule will bind, and here the blue one is a C3b molecule. So this is how the C3b is acting like an opsonin, and it is promoting the opsonization process. In the figure we can see C3b binds to microbial cells functioning as an opsonin. This is the antigen, and these are the C3b molecules, and these are the receptors of the phagocytic cell, and they are promoting the phagocytosis. So this was about the opsonization. Next comes complement fixation. So complement fixation, again the same. Here we will be discussing about the role of the antibody in complement activation. So I now only have told you in the pathway. In classical pathway, it was the only antibody which was recognizing. Antibody was recognizing the antigens, right? And then only it was initiating the complement cascade. And further the rupturing will occur. Further MAC complex and rupture of the membrane will happen. Rupture of cell membrane. So that is what the role of the antibody in complement activation. We can see that antibody antigen complex becomes bound by complement components in serum and is either phagocytosed via cells expressing C3 receptors or lysed as a result of pore formation by the complement component C7, C8 and C9. So again the same classical pathway which is using the antibody molecules to recognize the microbial surface. So here first step only is the antibody recognizing the epitopes, and then they are recruiting and binding with the C1 complex. This is the C1 complex. After that C1 in the C1s will be activated, and that will be cleaving C4 and C2, and they will be producing C3 convertase, and the C3 convertase will further convert the C3 molecules into C3b and C3a, and that will produce some C3 molecule again will bind and produce C5 convertase. So in next after C5 convertase, the C5 convertase again that will convert C5 into C5a and C5b. Again there is, after this C5b, it will bind with C6, C7, C8, and further at last they will form a MAC complex, membrane attack complex, which will be used for rupturing the cell membrane of the microbes or pathogen. So this is how the role of the antibody was, just to initiate the complement cascade, and further the rupturing of the membrane occurred. The last function of the antibody is antibody dependent cell mediated cytotoxicity. From here term only we can understand the cytotoxicity which is dependent on antibody. So if in a brief, if we say antibody antigen complexes are bound by Fc receptor or NK cells and granulocytes, thus directing the cytotoxicity of these cells toward the antigen targeted by antibody. What is happening here is that first the antibody will bind to the antigen, okay, and then the Fc receptor will bind to the NK cells and the granulocytes, and that will, after binding with those receptors, that antibody will direct the cytotoxic cells to further process the antigen, okay, and further there will be an apoptosis or killing of the target cell. So what is the definition of the ADCC, antibody dependent cell mediated cytotoxicity? Is the killing of an antibody coated target cell by cytotoxic effector cells through a non-phagocytic process characterized by release of content of cytotoxic granules, which can be perforin, granzymes, tumor necrosis factor and all, or by the expression of cell death inducing molecules. Okay, suppose this is the, again I will be showing you this, the antigen and the antibody is this, and they will be binding with the receptor of the any cytotoxic cells. Suppose this is the neutrophil, and after binding with this there is a signaling inside that cell, cytotoxic cell, has to release some mediators. So now they will release some mediators against the antigen, okay, and further there is a death or destruction of the antigen. So what are the receptors? Fc receptors are the trigger molecules for ADCC. Now different receptor, they will bind with the different immunoglobulin molecules, okay, like FCR, Fc gamma R, it will interact with the IgG. Fc alpha R, it will interact with IgA. Fc eta R, it will interact with IgE. So all these, they will mediate different signaling, and again this process will be same, ADCC will be happening. And what are the cellular effectors of the ADCC? What are the cells responsible for doing these? So a natural killer cells, it will be expressing the receptors for like CD16, receptors for IgG, then macrophage, monocytes, dendritic cells, then neutrophils, eosinophils, in the diagram also we can see the different cells are contributing. Suppose this is the target cell, and it is expressing its epitope. The antibody is recognizing. After recognition, it is that antibody's Fc region will interact with the any receptor of the neutrophil. Again, the same is happening with the NK cell. It is also interacting with the NK cell. Now, after interacting with these receptors, now there is a signaling inside the NK cell, and that will further release different mediators like perforin, granzymes, tumor necrosis factor. So, different molecules like eosinophil macrophage are there. So, let us see the mechanism of cytotoxicity. The same thing is happening. Antibodies bind to antigen or target cell. This is the target cell, and this is the epitopes which these target cell expressing. These are the antibodies binding. After that, primary NK cell recognize cell bound antibodies. Now the NK cell, they have interacted with this is the receptor of the NK cell, and this is the Fc region of the antibody, and this has interacted with the NK cell. Now cross-linking of CD16 triggers the release of perforins and other proteins. After this, there is a further cross-linking and further signaling will occur, and after that there is a release of perforins, granzymes for killing the target organism. Target cell killing occurs through the process of apoptosis, and there is an apoptosis process and target cell killing will occur. So in this way we have covered the antibody effector function, how these antibodies are so much important for functioning of different processes like neutralization, opsonization, complement fixation, antibody dependent cell mediated cytotoxicity. So this was all about the antibody functions. Thank you.